Okay, so let's move to the next speaker, who is Erika Pierattini, who is going to share with us the getting rid of PPCPs, some help from the plants, phytoremediation, and emerging contaminants. Please, Erika.
So thanks, everybody, and good afternoon. My talk will be about a novel way to deal with pollution from pharmaceuticals and personal care products. That is the use of plants to remove pollutants from the environment, and that is known as phytoremediation. So we have just a quick recap on what are pharmaceutical and personal care products. Of course, we have human and veterinary drugs, but also other substances of use and abuse, such as caffeine, nicotine, and recreational drugs. And also we have plasticizers and other additives and compounds that belong to cosmetics, fragrances, and detergents. So why these classes are of increasing concern? They are very important because they are continuously released in the aquatic environment, and often they are not efficiently removed by traditional wastewater treatment plants. And moreover, they can enter the food chain, for example, with the use of reclaimed waters for irrigation of fields. So the goal of phytoremediation is to use plants to remove pollutants from soil and water, and this may happen through several mechanisms. The pollutant may interact directly with the plant roots, and this can be stabilized at root level to prevent washing into the groundwater, or it may enter inside the plant and be translocated into the aerial parts, where it can be accumulated, and therefore we talk about phytoextraction. That is typical when the target pollutant is a heavy metal, or it can enter the evapotranspiration stream and be volatilized. The organic pollutant may also be degraded, or by the bacteria and the fungi that are associated to roots in the rhizosphere, or they may undergo phytodegradation inside the plant cell. And phytodegradation is, in fact, the most interesting aspect of phytoremediation. And the metabolic processes that the organic pollutants may undergo inside the plant cell have been widely described, and they are known as the green level model. The main processes that may occur have been grouped into three phases. The first phase is activation. So the organic pollutant is modified by the plant enzymes, for example, by hydroxylation in the cytoplasm. The phase 2, the pollutant is conjugated to biomolecules such as carbohydrates, while in phase 3, the pollutant is sequestrated in the vacuole or in the cell wall in order to reduce the harm to the plant cell. And these metabolic processes are not necessarily subsequent and may occur independently one from another. So which are the plant species that suit best for phytoremediation purposes? There are a lot of studies concerning various species, and mainly there are herbaceous species such as Phragmites, Scirpus, and Tifa, but also crops such as Brassica species, while amongst trees, the most used for phytoremediation purposes are Salix and poplar especially. And in the case studies that I'm going to show you, we use poplar because this is a plant that shows very important traits for phytoremediation, such as high grow rate, a large root system, large biomass production, and moreover, it has a great economic value for the production of wood, paper, and biofuel. And in particular, in our trials, we use a white poplar clone that can grow in vitro condition, and it's really performing in laboratory trials. All the trials were conducted in hydroponics inside a growth chamber, and we monitored the health of plants for the whole duration of the experiment, since it's very important that plants maintain good health for a successful remediation plan. And at the end of the experimental period, the plants were harvested and the various organs were extracted and analyzed with LC-MS using a 6-triple quadruple for target analysis. So in the first example I'm showing here is a trial with the antibiotic erythromycin, where we observe that this pollutant is mainly retained in roots, but it is also translocated in leaves and stems if the plants are exposed to the pollutant at higher doses for longer periods. And we also found some evidence of plant metabolism because, interestingly, the mass chromatograms of roots treated with erythromycin presented some additional peaks that share the same mass transition of the target antibiotic and that a retention time very close to the erythromycin 1. And in this case, separation was carried out on a Gemini C18 column using an electrospray source in positive ion mode. And we hypothesized that these additional peaks were epimers created by plant metabolism, but unfortunately, we could not further investigate them in this study. And an issue with dealing with phytoremediation are interferences from plant matrix, which are really complex matrices. And when performing phytoremediation trials, it's mandatory to have some plants grown in uncontaminated conditions to act as a blank in order to estimate both recovery and matrix effects on target compounds for LC-MS analysis. Also, other interferences may come from the laboratory equipment. And in this case, labeled compounds are really helpful. For example, in this case study, we were dealing with phthalates that are really ubiquitous in laboratory equipment. And so using labeled dialkyl phthalate as a marker, we observed that this class of molecules are very poorly absorbed by poplar plants, and they are mainly retained in roots. But we also found some traces of the deuterated dialkyl phthalate in leaves of both control and treated plants, and that show that poplar can, in fact, absorb these compounds directly from air, since the high volatility of phthalates is very well known. But labeled compounds are useful also when dealing with pharmaceutical and personal care products that are of plant origin, such as caffeine, for example. And the use of labeled caffeine, in this case, allowed us to monitor both absorption of this pollutant and the first step of its degradation process by demethylation into its metabolite. So there are theobromine and theophylline. And moreover, for the first time, it allowed us to discover presence of endogenous caffeine and both its metabolites in poplar. And we can see in this leaf mass chromatogram that endogenous caffeine was present both in control and treated plants, while labeled caffeine was present only in the treated one. And in this case, the separation was carried out on a Gemini C18 column with an APCI source in positive ion mode. And we found out also that endogenous and exogenous molecules undergo different metabolic pathways in poplar plants. And in fact, endogenous compounds are more abundant in roots. And the prevalent endogenous metabolite is theophylline. While once labeled caffeine is given to plants, the molecule is rapidly translocated in leaves and it's degraded in theobromine. And for the last case studies I'm going to show you, this case study was conducted using sodium dodecylsulfate as a model for detergent. And surfactants such as sodium dodecylsulfate are often used in phytoremediation trials in order to ameliorate the solubility of heavy metals in polluted soil and therefore enhance their uptake from plants. So we set up a trial of sodium dodecylsulfate in combination with zinc. And as we observed with phthalate, the dodecylsulfate molecule is highly retained in roots. And also there is a high contamination of also controlled plants from this detergent. And also we actually recorded an increase in zinc uptake as a consequence of sodium dodecylsulfate co-treatment. Our poplar plant manifested browning and subsequent falling of leaves. There was a stress symptom caused by the sodium cation of the sodium dodecylsulfate molecule. which induce a so stress-like phenotype. So we really should consider to always perform preliminary laboratory screening before starting a field phytoremediation project in order to select plant varieties that are suitable, more tolerant, and more effective against the various pollutants that are present in a site of interest. So thank you, and I'm ready for questions.
Yeah, thank you, Erika, for your nice presentation. And again, I'm asking all the audience if somebody has some questions. Pietro.
I have one.
Yeah.
Yes.
Probably is a silly question. So in the case where you were showing the antibiotics, so if I have some antibiotics in my hydroponics, how much of this is uptaken by the plant? I mean, is actively pumping the antibiotic out from the water, or is a simple system of since it's collecting water, it will also collect the antibiotics? Is it clear?
Yeah, I think if the question is how can the plant absorb, is an actively absorption?
So, yeah, it's an inactive process. So basically, if I leave there the plant, it will keep on collecting antibiotics, or is simply a diffusion? So basically, so it's in terms of phytoremediation. So yeah. how much should I grow the plant there to get rid of the antibiotics or something like
so this? In that case, the experiment lasts for 4 weeks, and every week the solution was renewed with new antibiotics. So in fact, the plants had a lot of quantity available, so they continuously pump the antibiotic inside, or at least, of course, they don't have any dedicated plant transporters, you know, but sometimes in some way the molecule enters and is also translocated to the other part when the concentration is quite high and for a long time.
Okay, thanks.